Today we will introduce the sixth tool of HereIm, morphological analysis of fruit flies mitochondria. In this image, we can see the fruit fly's germline stem cells. After skeletonizing, we can analyze its morphology. First, let's explain what skeletonization is. Skeletonization shows a tree-like structure here. After skeletonization, we get 3 branches, 1 junction, and 3 terminals. By counting the number of branches, junctions, and terminals, we can quantify its complexity. How to perform this analysis on mitochondria? First, here is an image for you. It shows a fruit fly ovary germline stem cell. The green part represents our mitochondria. By selecting cells and adjusting the threshold, we obtain this mitochondria mask. Next, we skeletonize the mitochondria mask and get the skeletonized image. This image overlays the skeletonized image with the original raw green channel, which is the mitochondria channel. Let's continue to see how to use this tool. Now, let's look at the table. This was written by Dr. Hwei-Jan Hsu. How to use this tool? Here are some instructions below. Also, we can click on the GitHub link. There are descriptions there as well. Let me briefly explain. After running the script later, we will get 2 files. One file stores mitochondrial complexity. The other stores its volume and intensity. Now, let's go over how to use it. First, open this ImageJ file. Then press Ctrl A. Next, copy it. Then, go to ImageJ. File. New. Then Script. Paste it here. Let's first briefly explain what the script does. Here is a simple flowchart. Now, let me first show you all our original image file. We will first use this original image file. Select the location of the cells. Then obtain this mito mask. Then, proceed with the analysis of its intensity and volume. And then, in order to understand its morphology, so, we conducted skeletonized analysis again. That is this protocol here. The different colors here represent that he belongs to different skeletons. This image is the confirmed image. Let us know if the skeleton we drew is the same as what we expected. Now let's get back to our script. First close the previous file. Select the image, and click the Run button. It will require us to define our cells. On this composite image, manually select the cell. Press this selection tool. Then, select one cell. Then press OK. Next, we will use the threshold to define our mitochondria cell mask. Simply cover all the green areas. You can slide along the z-axis to check. Then click OK. That will be our cell mask. It will then perform a series of analyses. How do we check the results? It saves a file like this in the original directory. OK. Like mentioned earlier, we will have 2 summary files. One is the branch summary, meaning whether something is part of the skeleton. From this we can tell how many branches it has, and for each branch, the number of junctions, the endpoints, and the junction voxels, plus average lengths, and so on. The other files house the mitochondria's volume and its intensity. This is its intensity value. All right. The other files are the ones we showed earlier. We save them. If you have any questions, feel free to email us. That's all for today's video. Thanks everyone.